right, everybody, let's get started. Thank you so much for coming. I am Karen Pierce, professor in the Department of Neurosciences at UC San Diego. We're all here today to learn about genetics. And when the human genome was first sequenced in 2003, there was a collective excitement about the world. There are now so many new possibilities in understanding risks for certain diseases and disorders, for understanding how genes interact with the environment and influencing health outcomes. And my personal favorite is the potential for precision medicine, where treatments and medications can be tailored to an individual's genetic profile. Today, we are so lucky to have Dr. Jonathan Sebat, professor in both the Departments of Psychiatry and Cellular Molecular Medicine at the University of California, San Diego. Dr. Sebat is also the director recognized scientist. He has been how our genes influence the manifestation of certain disorders, such as schizophrenia and autism. And with that, I now bring you Dr. Jonathan Sebat.
Thank you, Karen. It's great to be here and understand mechanisms. And that understanding has knock-on effects on other aspects of autism research, including studies of environmental factors. And so I think we can, and there's a great deal of progress in these areas. So I think that I can really help to kind of highlight what we actually know. And I'm going to start with, so you can see from my talk title that we're not just talking about genes. We're talking about genes and environment. We're talking about the causes of autism, something that's in the news a lot right now. And it's something that is not a mystery to us. It's something we actually have learned a great deal about. And it's good to communicate this to each other and to the public. I have no conflicts of interest to disclose. The research that I'll discuss today was supported by the National Institutes of Health, the Simons Foundation, Autism Speaks, Autism Science Foundation, the Beisters, the Dickinson Foundation. None of it was supported by the pharmaceutical industry, I guarantee you. I'm going to start by reading some of my slides because I have a story to tell, the story of James, right? So let's meet James. James started life as a happy, thriving baby. For his first year, he was happy and social. He hit his milestones on time. His babbling turned into simple words like mama and papa. He made eye contact, responded to his name. His motor function was right on track with crawling and pulling himself up to stand. And he would respond to his parents' attention with joy. James' parents knew that this was good progress and that he seemed to learn something new every day. At around 12 months of age, James received his routine MMR vaccine. That evening, he had a fever, he was fussy, and the doctor said, "Don't worry, it's a normal vaccine reaction." But over the next few weeks, he stopped speaking the words that he had learned. He no longer responded to his name. He stopped pointing, waving. He became less interested in playing with his parents and with other kids. He avoided eye contact. He did hand flapping and rocking and rolling back and forth. And he became upset by loud noises or by changes in his routine. So it was clear now, he was not the same social, happy baby that they had known. James's parents really couldn't shake the feeling that something had happened following the vaccination. This can't be a coincidence. James was clearly now not neurotypical. He had social and communication difficulties. He had rigid routines and unusual behaviors. And after months of this, a developmental pediatrician diagnosed James with autism spectrum disorder. And James was not alone by any means. James' parents wanted to find answers, and they looked for answers. In online communities, they found other parents that had eerily similar experiences. She was so social, then one day it was like she wasn't there anymore. And those other parents had also noticed that those symptoms emerged after the 12-month MMR shot. So how can so many people having the same experience be wrong? So the CDC listened, and the stories of the children regressing after their 12-month shots really kind of pointed to the MMR vaccine as a potential cause. And so large-scale research studies were launched. The CDC, NIH, WHO did multiple large-scale studies in the US and Europe. Those studies found no link between the MMR vaccine and autism. 2004, the Institute of Medicine reviewed all the available research, and their meta-analysis concluded that the vaccines were not strongly linked to autism. And large subsequent meta-analyses reaffirmed these findings. Autism rates are the same in children who are vaccinated as in children who are not vaccinated. So what is going on? Autism has an early onset. It frequently emerges after the vaccines are administered. 20 to 30% of families report that the child acquired skills by 12 months of age and then lost skills after the shot. And the prevalence of autism is rising. So this is not a solved problem. There's a lot we still don't know about the causes of autism. But it's not a mystery. Autism is a complex mental health trait. Like many other complex traits, has an influence from both genes and environment. And the genes are clearly a major factor. And this is not new, right? Since the 90s, we've had twin studies that was able to show us that genes are a big factor. So what I'm showing you on the left are what you see in studies of twins when you look at the concordance rate in identical twins and fraternal twins for a disease that's 100% heritable. So if you have 100% heritability like you do in Huntington's disease, then identical twins will be 100% concordant for the disease. If one twin has Huntington's disease, the other twin will have it 100% of the time. And if they're fraternal twins, which are basically just siblings that are born on the same day, then if one twin has the Huntington's disease, there's a 50% chance that the other twin does too. It's essentially dominant, meaning that it's on one of mom or dad's chromosomes, and there's a 50% chance that it'll be transmitted, right? So you have 50% concordance. And then you see exactly the same thing in siblings that you do in the fraternal twins. That's what 100% heritability looks like, particularly with a dominant trait. And now let's look at an example of a mental health trait that's very heavily influenced by the environment, PTSD. So post-traumatic stress disorder, you have 30% concordance in the identical twins, 15% in the fraternal twins, and 15% in siblings. And that gives us an estimate of about 30 to 40% genes, 60 to 70% environment. So PTSD, unsurprisingly, has a major environmental trigger. So what about autism? The twin stories in autism are really looking a bit more like Huntington's than PTSD. You have 70 to 90% concordance in identical twins, 30% in fraternal, and 10 to 20% in siblings. This gives you an estimate of roughly 80% genes, 20% environment. Now, twin studies, they have lots of their limitations. We can geek out on whether they obey all the assumptions about shared genes versus shared environment. And feel free to at me on Twitter. We can go all day long about heritability estimates of various different types. But regardless of the details of twin studies, it's undeniable that genes are a major factor in autism. That's very clear, regardless of your limitations of twin studies and variability in what you might come up with as an estimate. It's very clear that genes are a big factor. So, okay, Einstein, where are the genes then? So I'll do a quick little bit of science history for you. In 2007, Mike Wigler and I proposed an alternative hypothesis that hadn't been explored in genetic studies before. We saw that most autism was sporadic. It didn't run in families. And so we hypothesized that, well, then let's look at the genetic factors that are sporadic. So we hypothesized that, in fact, autism was caused not by things that run in the family, but by new mutations that occur spontaneously in the child. That became a paper published in 2007 by Mike Wigler and I, where we scanned the genomes and looked for mutations with the genome scanning tools that we had at the time. So this panel A that you see on the screen is a deletion that was detected in one child with autism. And you can see that there's a loss of a copy of a region of genes in the genome. That's the blue plot. And the green and the red plots are mom and dad. And you can see that mom and dad do not carry the same deletion that the child carries. This is a mutation that occurred de novo in the child. It was not carried by the parents. And that deletion impacts a region of the genome that contains a number of genes. So de novo mutations are strongly associated with sporadic autism. And when autism runs in the family, meaning you have multiple siblings with autism, we see weak associations of the de novo mutations. So it is a sporadic mutation that's associated with sporadic disease. Now, the mutations that contribute most strongly to autism are, in fact, those de novo mutations that are not inherited from mom and dad. They're not the only thing that contributes to autism. There's actually a variety of genetic factors that influence. So you have the polygenic risk. This, just imagine your family as just one part of an enormous family tree. And the variants that are really ancient, the variants that have been present in the family since the beginning of time, are what we call polygenic risk. These are ancient variants that float around in the population. And they nicely shape our human traits, but they do not have extremely large effects and large and deleterious effects. They have small effects. And then there are other mutations that occur a little more recently in your family tree that haven't had much time to undergo natural selection in the population. Those types of rare variants will be relatively recent, and they'll carry larger effects than the ancient variants, because they haven't had time to really undergo natural selection in the population. And then, of course, there are 100 new mutations in every child who's born. And what our study showed is that this is where the big genetic signal is coming from in autism. It doesn't stop there, of course. We've also shown that about 5% of these de novo mutations are actually not occurring in the sperm or the egg, but they're actually occurring in the developing embryo as well. So there's roughly 5% of these that are actually mosaic in the infant, meaning that it's not present in all of their cells, and it wasn't present in the sperm or the egg. And other colleagues here at UCSD, like Joe Gleason and Chris Walsh at Harvard, are doing more studies on the mosaic variants. Those are a tough nut to crack. It's a little harder to do those at a population level and show their association. So really, the de novo mutations has really been where the major signal has been coming from. So that has ultimately become the main approach for finding autism genes. And we have to give credit to the pioneers who really made this possible. Really, we would not have 100 genes right now if it was not for Jim and Marilyn Simons. The Simons Foundation ultimately initiated the recruitment of trio families at large scale, and this DNA sequencing at large scale that ultimately led to the discovery of the genes. And so what I'm showing you here on the left is the results from 2020 of the more recent Autism Sequencing Consortium study that's using the Simons collections that were created. And that study found over 100 genes. And these genes, they're not just little markers in the genome that are maybe suggesting that something is there, right? Like you might have found from a genome-wide association study or a linkage study. These are causal variants. These gene mutations have profound effects in patients. They cause epileptic encephalopathy, intellectual disability, significant, learning and social impairments in children. When you bring all the children together that carry the same gene, you can see very similar kinds of clinical features within them. So each of those genes themselves has very strong effects. These are not little flags in the genome that are maybe pointing at something. These are causal variants. And when you put those same variants in mice, you get mice with significant neurological and behavioral phenotypes that you can see there. And so they're very quickly in these types of studies of de novo mutation go very quickly from discovering a gene to really defining the syndrome of the gene and translating very quickly in the neuroscience lab into an understanding of how that gene affects brain development. So these genes are not just a risk factor. Each one of these genes is a part of the autism spectrum. So as these new genes are identified, and as more and more of them are identified, the success rate of clinical genetic testing improves. So I'm showing you a 2015 paper from my colleague Steve Scherer at Sick Kids Hospital and University of Toronto, where they take clinical microarray technology and whole exome sequencing combined and ask the question, in a sample of idiopathic autism, how often do we get a positive genetic finding where we are confident that there is a genetic factor in this child? And the diagnostic yield that Steve got from that study was 30%. So 30% of kids with autism have a factor that's diagnosable with clinical genetic testing. You could take the general population of autism in the community right now, bring them into clinical genetic testing, 30% of them will get an answer from clinical genetic testing. It does vary, of course, by population. And it varies based on whether the kid's in mainstream school versus having severe learning disabilities. It varies a lot depending on who you grab. But if you grab everybody from the population, you get a significant number of positives from clinical genetic testing. So Mike Wigler looked at some of these results and started to ask himself, okay, we have enough data now that we might actually be able to look and do some modeling to estimate what are the total number of genes out there, and when, how much of the diagnostic yield do you think we might get to, based on how many of these genes are starting to show up in multiple kids, the same gene over and over again. You can start to estimate, what are the total number of genes. So Ivan Yosifov and Mike Wigler got an estimate of, so the blue plots here are the observed frequencies. So just the observed frequencies of positives in the population are roughly about 30%, as you can see here on the blue. And it's greater in females than in males. So your diagnostic yield is better with girls on the spectrum than with boys on the spectrum. Also, when you do the modeling to figure out how much we might get to in the future, the models give us an estimate of about 60%. So we might get to about 60% diagnostic yield if we had all the autism genes, which we don't. He can also estimate roughly how many genes those are. And the estimates comes to about 400. So again, these are just models, right? So they have their limitations. But according to the model, about 100 genes, and so we're 25% of the way there, meaning 20% of the genes undiscovered. Right? So the genes that are involved are genes that regulate other genes and that are expressed in early fetal brain development. And so essentially they're genetic switches that control fetal brain development. And so you have these things, the little blue circle in the center of the cell is the nucleus where all the gene regulation happens. And that's a lot of the action in the autism genes are right there in the nucleus, the regulation of fetal brain development. And then after that, you also have cell signaling factors like Wnt signaling, MAPK signaling, mTOR. These are pathways that regulate the growth and differentiation of neurons. And so you're starting, the little pieces of the puzzle are starting to fit together into pathways that regulate fetal brain development. And then on top of that, you have aspects of brain function that are directly involved in learning and memory, like synaptic neurotransmission. So just from a list of genes, you get a lot of clues even before you even go into the lab. And of course, again, this list of 100 genes are going straight into the lab as we speak, and there are extensive studies now underway to understand how these genes do regulate brain development. But we're here to talk about genetics and environment, and so let's kind of circle back to James. Remember that this was a kid who lost his skills right after the vaccination. So what's going on? So there have been studies looking at the diagnostic yield in autism. And of course, you can apply the same type of approaches to people with regression. So this is a study by Katrina Williams in Melbourne, where she specifically applied clinical microarray and whole exome sequencing to cases of autism that gained their skills at roughly 12 months and then lost them. And those individuals with regression had a very high diagnostic yield from genetics. So the exact percentage here, I wouldn't hang my hat on this 50% number. This is a sample of 99 cases. No one should ever hang their hat on a sample of 99 cases of anything. And they're not necessarily a representative sample of autism. So a lot more work needs to be done. We need genetic studies of regression. We need genetic studies of suspected vaccine injury. And the genetic findings will be illuminating. The genetic findings will allow us to understand mechanisms of disease in those individuals, and can start to help us to understand a bit more about genes and environment. And then, of course, I think the families just want answers, right? The families want answers. And so if that answer is a gene, then that's a good thing. Now, as I said, there's the limitations that is that a representative sample? So who knows what the actual numbers are? We really don't have good studies of idiopathic autism regression. We still need those. But it's already clear from Katrina's findings that a lot of the genetic findings that she's getting are the 100 autism genes. So we have MECP2, IQ-seq, KMT2C, SHANK3, SCN1A. These are the autism genes that are coming out of the exome sequencing studies that the Simons Foundation has been funding. So I think it's a reasonable conclusion that there is a genetic basis to regression. So, like I said, whether it's gene or environment or both, the gene gives you an important clue. It's an important piece of the puzzle that allows you to understand the whole bigger picture. So let's focus on one gene here, SCN1A. So SCN1A causes Dravet syndrome. And we can specifically look at Dravet syndrome and how they respond to vaccines. Parents are not imagining things. Infants with SCN1A epileptic encephalopathy do show a peak of seizure onset within 48 hours of receiving the vaccine. So you are a kid with Dravet syndrome, the onset of your first seizure hasn't happened yet. You get your vaccine, you get a fever, and then that fever can precipitate a seizure, sometimes the first seizure. And so disease onset correlates perfectly with the vaccine, and the onset is precipitated by the fever. So parents are not imagining things. Children with high genetic susceptibility do show an adverse response in that 48-hour period after the vaccine. They're not wrong. But the cause of the seizure is the epileptic encephalopathy. It's not the shot. If you actually compare the actual effect of vaccination itself has no effect on long-term outcome of Dravet syndrome. So the SCN1A causes the same epileptic encephalopathy with or without the shot. So you have a transient effect of the shot, and you have a high genetic susceptibility that makes you prone to seizures. Your long-term developmental outcome is determined by the gene. It's not determined by the vaccines. But the parents are not imagining things. So the scientists on one side are saying it's not the shots, and the parents on the other side are saying, yes, it's the shots. You're both right. The scientists are right. It's not the shots. But the parents are not imagining things. There really are transient reactions to shots that are unique to highly susceptible individuals. But wait a minute. Genetic testing in James was negative. Now what? Well, regardless of gene or no gene, there will always be a correlation between autism and the vaccine schedule. They are inextricably linked temporally. Early developmental milestones in kids, the early ones in 6 months are really basic motor coordination, begins to sit up, responds to his name. And so, these are very, very basic types of milestones. And those are not the milestones typically that are failed to meet in autism. The milestones that are really diagnostic of autism spectrum are the milestones at 12 months of age, uses gestures, points, responds to spoken question, pulls up and stands, says mama, says dada. These are the milestones that are diagnostic of developmental delay. So regardless of the trajectory of the child at the 6-month time point, the diagnosis of autism comes when you fail to meet the milestones at around the 12-month period. And failure to meet milestones correlates perfectly with the first MMR shot, which is done at the 12-month checkup. So you will never eliminate the correlation between MMR and vaccines. MMR and autism. It will always be there. It will never go away because it's just correlated in time. The developmental milestones and the shots are on schedule. They're on the same schedule. So, okay, regardless of the vaccines, what's going on with the prevalence? Autism prevalence is rising. The prevalence is now 1 in 36. Genetics does not cause an epidemic. Explain this. Not the vaccines. So you can look at examples where where thimerosal was removed from the vaccines, and before and after removal of thimerosal, that prevalence kept right on climbing. It didn't stop. In Yokohama, there was a 2005 study showed that you remove the MMR vaccine entirely, the prevalence kept right on climbing. There are factors that we know contribute to the rise in prevalence without question. We've been able to demonstrate them pretty clearly now. So a 2015 study showed that changes in the diagnostic criteria explained about 60% of the increase in prevalence up to 2015. An earlier study found that about 50% of the prevalence increase up to 2007 was attributable to diagnostic substitution. A kid who receives an autism diagnosis instead of a diagnosis of intellectual disability, whereas maybe earlier in history that person might have been given a diagnosis of ID. Now they get a diagnosis of autism as a main code that goes into the health record. Now, we can control for these things. So the Autism Developmental Disabilities Monitoring Network run by the CDC controls for these factors, right? So you control for diagnostic criteria. The diagnostic criteria are not changing. And the ADDM is still showing a rise in prevalence. It's the ADDM that's telling us that the prevalence is rising. So now that you're controlling for diagnostic criteria, how do you explain what's going on? Well, ascertainment that I'm talking about is not just that. right? There's a lot more to it. So, and it's not just awareness, scare quotes. The ADDM is a network itself, and that network is connected to a broader network of healthcare facilities, public schools, special ed programs, therapy providers, developmental services. And each one of those is connected to a network of people and sources where the kids are coming in from. And so you have this network upon networks of ascertainment that's capturing the prevalence of autism in the community. And so even a modest increasing awareness globally can affect all of these things in terms of what gets referred into the system. And then, of course, changes, updates to clinical guidelines, changes to medical practice, changes to what clinical departments within the hospital see autism and how often they refer. How often does occupational therapy refer a child from their department to get evaluated for autism? There's all kinds of elements to the system that can all get tweaked in small ways that will increase the funnel into the ADDM. So the ADDM was not designed to find the causes of autism. The ADDM was not designed to tell you whether the prevalence is really increasing or not, or is it just diagnostic or whatever, right? So that's not how it was designed. They were designed to track the prevalence and to see differences in prevalence between California and Maryland and things like that. So there is still, like I said, it's highly complex how the ascertainment part can work. So is that what you're saying? That's it? Prevalence is not really increasing? Is that what you're saying? It's not what I'm saying. I'm saying that ascertainment is a big part of it. It might even be the biggest part. And any study that's looking at links between vaccine and autism has to prospectively incorporate the ascertainment component, and has to understand how that's influencing the study. Any scientist that's looking objectively at the data has to say, no, I would say, let me take a step back. There's a lot of scientists out there who would look at this result and say, come on, all right? I mean, we have resources. We have to be careful with our resources. Let's go after the most promising directions. Let's not invest resources into things that are already, not promising. And that's a general attitude in science. You have to do that. You have to economize your resources. Where are you going to invest your time? Where are you going to invest your money? So that general practical approach that scientists have would lead them to steer away from, environmental factors. Genetics plays a big role, and I say no. I'm a geneticist, and I'm telling you, don't do that. So the heritability of autism is not 100%. There are many environmental factors that have been implicated that are increasing over time. Survival of preterm births and NICU admissions are increasing. From the 1980s, the survival rate of kids in the NICU was 50%. The survival rate in the NICU is now 90%. There's almost a twofold improvement in survival of kids who are struggling in the NICU. And this changes the population. There are kids in the population now who didn't used to make it, who make it now. Parental age is changing over time. The proportion of men who have children after age 40 has increased since the 80s from 4% to 9%. So more than double the number of fathers who are older. Air pollution has a reproducible correlation with autism. It's not well understood. Also, it's increasing over time, not everywhere. So it doesn't explain the increase in LA because the air pollution has actually decreased in LA since the 1980s, but it's increased in Denver. And then, of course, there's a variety of other things going on here, like epigenetic signatures of environmental exposures. I can look at the methylation on your DNA, and if you're a smoker, we can tell you're a smoker, right? Or we can actually predict your age based on the methylation in your DNA. So there's all kinds of signatures that the environment leaves on the DNA. That's a tough nut to crack. Those studies are mostly in animal models. It's a little harder to link it to human, but these are studies that have to be done. We have to do that in human, and we have to try to figure out ways to actually explain, see, demonstrate a role in health in humans. Again, each of these things is an interplay of genes and environment. So when you say environment, you are always talking about genes in some way, shape, or form. So let's look at preterm birth, right? Preterm birth has a major increased prevalence of autism. Kids that are born preterm have kids that were admitted to the NICU and then discharged from the NICU have a dramatically increased risk of autism and other developmental disorders. Preterm birth and neonatal distress is itself a mix of genes and environment. Our colleague, Stephen Kingsmore at Rady Children's Hospital, has been implementing whole genome sequencing clinical diagnosis of children in the NICU. And 10 to 30% of NICU admissions are later determined to be genetic conditions like Down syndrome, Noonan syndrome, skeletal dysplasia, Charge syndrome. So the NICU is a heavily, environmentally loaded group who was born preterm before the lungs have fully developed, before the brain has fully developed. But it's not just the environment that's going on here. You also have genetic susceptibility that actually contributed to the preterm birth in the first place and contributed to the NICU admission in the first place. So the increased NICU survival is a factor that may contribute to increased vulnerable fraction of the population. Talked about parental age. So fathers and mothers who have children over the age of 40 have a roughly 1.5-fold increase probability of having a child on the spectrum. And studies in 2012 showed that fathers, as they age, have increased de novo mutations. So de novo mutations being the biggest genetic signal in autism, it doesn't look like a coincidence that the older fathers are accumulating roughly 1 to 2 new mutations per year as they age, that the increased number of mutations and the probability of disrupting a gene involved in brain development increases as men age. Women to a much lesser extent. It's really a male thing. Fathers accumulate mutations, mothers to a much less of an extent. So there you go. Wow. Okay, it's an environmental factor that actually has a genetic mechanism, and a simple one at that, de novo mutation, right? Not simple. So parental age effects are actually a highly multifactorial genetic mechanism. So a study from my lab a couple of years ago showed that every single genetic factor that we can measure that influences autism susceptibility has a correlation with mother's age or father's age. So you have the de novo mutations on the left are associated with father's age. They accumulate in the sperm of older fathers. Oh, you also have inherited mutations that can be carried by mom or dad. When dad carries the inherited mutation, he's younger. And you have polygenic risk, that those ancient common variants that have been floating around in the population that have modest effects on you, modest influences, those are correlating with older parents, and particularly for moms. And so you have all of the genetic factors are correlating with age, and they're highly sex specific. So, and we actually understand the genetics is teaching us how this works. So by looking more carefully and dissecting the different correlates of these factors, what we can see is that all of these genetic factors are tied to learning and education in the parents. So if you look at the correlation of father's age and mother's age, and then you look at their effect on educational attainment, what you can see is that the genetic predictors of educational attainment are ultimately correlating very strongly with their effects on mother and father's age. And they all have some direct influence on autistic traits. So here you have a polygenic score that's a predictor of high education, and it directly influences autistic traits. Mom's age that's a genetic predictor strong in moms. Mom's age is strongly correlated with dad's age, and mom's age is strongly correlated with her level of education. So moms with PhDs have children later, and so you can see that the genetic predictors of high education are correlating with age in a logical way. And so mom's age is correlated with dad's age. And so the older father has the increased rate of de novo mutations. And so you have a sprinkling of additional susceptibility on top of what's already there. So almost by definition, the parental age effect is a multifactorial mechanism, even within the same family, potentially. So what the research is telling us is that we have good evidence of genes and environment and the combination of all of the above. And there's good evidence of it. We have really good, clear evidence of de novo mutations, inherited variants, and then polygenic risk. And these are factors that have a direct influence on brain development and cognitive traits in the general population and in autism. And there's, of course, environmental factors that are also influencing probably the same mechanisms and pathways. They're a little harder, again, like I said, they're a harder nut to crack. It doesn't stop there. You have genetically determined environmental exposures, like preterm birth. And preterm birth is itself an exposure that strongly increases your risk. So you have environmental factors that are triggered by genes. You have environmental factors that feed back on the genome, parental age, increasing the number of mutations and increasing your genetic risk. So you have both bidirectional interactions between genes and environment that are further kind of adding to the influence that's going on here. So it's not simple, it's complex, but it's not a mystery. So I think a message that I think I've managed to convey is that it's important to study the environment, right? Now, the scientists that again, vaccine skeptics go online and they might talk to a scientist, and that scientist will say, "I'm sorry, vaccines don't cause autism. Try again." And I would say that message is not the right message to send, right? In fact, the genetics, if anything, the genetics is teaching us important details about how genes and environment interact. So the real message here is not try again, or you're just looking for something to blame. That's not the message here. The message here is we hear you. We think we kind of understand it. We don't know it. We don't know everything. So the environment is worthy of study. It is vast, constantly changing, hard to measure, highly intertwined with genetics. So we need studies of environmental factors. We need them. And incorporating genetics into the design is crucial. In fact, it's essential. You won't be able to understand the mechanisms very well without it. So really, and I'll add, there'll be one last little vignette that I will add to this story. If vaccination has a causal role, genetics could actually provide evidence to support that. So going back to our paper from a couple of years ago about multifactorial inheritance of autism, right? There's some very nice clues in there. So the panel A that you see in front of you right here, these are boys and girls on the spectrum, and the hashed plot is how much of the polygenic risk there is in that individual, and they're separated based on whether they have a de novo mutation in a important gene or not, right? So the boys who have a de novo mutation have less of the polygenic risk, and the boys who do not have the de novo mutation have more of the polygenic risk. So again, if you have more of one thing, you don't need as much of the other. It's a typical kind of liability threshold model, they call it. The girls show the same effect, although in this particular paper it wasn't significant. But this is a reproducible effect. If you combine the girls and the boys, and you look at all the genetic factors, you have a very nice, apologize for geeking out on a data slide for a minute, but basically this matrix that you see on the right is looking at all the common factors on top and all the rare variant factors along the left side, and they negatively correlate very nicely. Each rare variant is negatively correlated with the polygenic risk. So that's a phenomenon that is now known and it's a useful tool in genetic studies. So you can see two genetic factors that are independent of each other will be negatively correlated. So two causal factors in your population will be predictive of each other. And the same holds true for any causal factor. So if vaccines had a causal role in autism, you will see it in the genetic data. You could see a negative correlation between an environmental exposure and a strong genetic predictor. So vaccines have been in the news. The media has reported recently that the CDC plans to do a study of vaccines in autism. And I think scientists are justified in saying, come on. Okay, it's not where the signal is coming from. It's not where the clues are leading us. Let's follow where the clues are leading us. So I have a message for RFK Jr. And for the public. People on the spectrum, their families, caregivers, and the scientists, they all want the same thing. We all want answers. We all want to understand what's going on. We have some answers. We got 100 of them and counting. But there's still a lot more that we don't know. So we need to understand it. We need to understand the mechanisms. So, and those mechanisms include environment. You can't get away from the environment. The genes affect the environment. The environment affects the genes. You really have to focus on the environment and the genes at the same time. But a new study of vaccines and autism is not that, right? So it's as if leadership in the CDC would be completely ignoring all of the science. So I and it's at this point it's just media reports, right? For all we know, it was just a conversation in the men's room. Like we don't really know what's going on. So we don't know what's going on. But what we can do is we can make our voices heard. We can tell the government, we can tell the public, this is how it works. And here's what we know, and here's what we don't know. So what the media is reporting that the CDC would propose to do is destined to be the most politicized research study in history. That's not how you build trust. Let's design a study of environmental factors, one that looks at a broad array of exposures and one that carefully accounts for genes. Let's work with the community to ensure trust in the results. Let's write all the code in advance. Let's let the data just come in and hit go and let the results just spit out the other end. And everybody take a look at the results. Parents, vaccine skeptics, scientists alike, let everybody just look at the results and then accept them and go get a beer. So we need to ensure trust in the results, and we want to continue moving forward. That's the goal of science. And that should be the goal of our funding agencies. Thank you.
Hey, thank you so much, Jonathan. That was really fascinating. We talked a little beforehand, and he described to me kind of this really sweeping and really powerful lecture that he was going to be giving, and you delivered as promised. It was really provocative. I really enjoyed it. I have, if people have some questions, please share them in the chat. Don't leave because I have a quick poll, but just a quick conversation. Since I'm the PI of one of the Atom Network sites here in California, one of the biggest factors that you didn't mention on your slide is just a simple thing of the digital revolution. Historically, when the Atom Network was trying to figure out, okay, how many people with autism are in this school or this hospital, it used to be paper records. And so the simple mundane truth is now it's digital. We can just access health records like that. So on top of the two reasons that you mentioned, which is increased awareness, and also people used to be called intellectual disability, now they're called autism. The biggest factor really is just way better access to data and digitization of medical records that allows for really rapid and accurate counting. So that, for me, as a scientist, is the hugest factor. Also, I totally agree with what you're saying, that we do want to understand some environmental factors related to autism. But at the same time, I really loved in the beginning of your lecture how you showed that monozygotic twins who have the identical DNA, concordance rates are between 70 and 90%. So to me, that's an enormous, loading for genetics in the outcome as it relates to autism. And so, I think that it is important to understand. And I loved your slide talking about the timeline, how vaccines happen at exactly the same moment that these developmental milestones happen. But I worry that people, might start, it just depends on how the message is sent. I think your message is one that's really healthy in trying to understand really this interplay between environment and genetics and how people with a genetic liability, of course, are going to have a bigger reaction than someone who doesn't. But that doesn't mean it's causal. And so I feel like I worry the public could just sort of hear the buzzwords, vaccines and autism, and then I worry about rates of vaccination going down in our country and what that impact could be. But I like the spirit of the way that you're talking about these things in a really healthy and productive way. So, but I don't want to hog up this conversation. So we have somebody who does have a question. It says, "Do you believe folic acid can help these children in their brain development?"
So diet, I mean, I limited my gene environment examples to just a few, but obviously diet is massive, like massively intertwined with genetics, right? So if you do a phenome-wide association study, if you take any one of these autism genes and then go into big biobank data and ask what phenotypes in the general population are correlated with these, guess what comes to the top of the list? Diet. Whether they eat cereal in the morning or whether they like fish. Like these kinds of things are highly connected to your genetics. Like those are in fact, we like to filter those phenotypes out. We get annoyed. We get annoyed by, I don't care if the kid eats fish or not, right? But in fact, it's highly correlated with genetics. And so, and of course, folic acid is an obvious one because, there have been many, many studies that talk about neural tube defects and the therapeutic value of folic acid when there are neural tube defects. My colleague Joe Gleason has a really nice study recently just showing that one of the deletions that I actually showed in this talk actually increases your risk of neural tube defects. And folic acid may very well be therapeutic in that group. I don't know. Again, the data on folic acid and outcome in autism. Again, these are trials that can be done. If anything, you would expect that the therapeutic value would vary between different genetic subtypes in the autism spectrum. I think that's a fair assumption. So number one, yes, folic acid would be a worthy thing to study. You need trials and you need genetics. You have to understand you have to understand who's responding and who's not.
Okay, great. We've got a couple of other questions. Should we invest in practices to protect the brain health or gene mutation?
That's a very broad question. So I could go on for hours. So the protection that you're looking for is genetic protection on the one hand. Again, that it's we have modest increases in our susceptibility due to age and other factors that affect the genome. So it's really, I made a conscious choice to have my kids while I was still a postdoc. I did. It was a conscious choice we had, right? I was like, I should probably wait till I'm assistant professor, but I didn't, right? I said, we were doing this research and I said, let's do it now, right? So, or maybe it was actually Lily who decided, whatever. But that's a personal choice, right? The effects are modest in terms of the genetic susceptibility and the effects of age and these other factors. So it's not like the clock is ticking. You have to do it today. You have time to work with. But these are personal choices in that respect. In terms of public health, is there something we would want to do for genetic protection or for other types of protection? Again, the research has to be done. Right? So I can't really answer the question with a public health recommendation or a clinical guideline because all of these things have to be based on evidence-based medicine has to be based on evidence. So the studies have to be done to really figure out mechanisms and things that should be done at a public health level or at a level of healthcare.
Well, thank you so much. I thank you to everybody who answered questions. We can try to email you back. I just want to do a quick poll. Don't leave. So I am going to launch this poll for you. If you could just answer these questions, that would be fantastic. Thank you, everybody. Really great discussion. And thank you again, Jonathan, for a really fun and thought-provoking presentation. It was fantastic.
It's my
So
pleasure. I had fun.
thank you, everybody. Okay, and what's amazing is the overwhelming majority of individuals do not have a child with autism, and everybody loved this presentation and got a lot out of it. So, all right, thanks everybody. I think everybody's mostly answered. All right, have a great afternoon.
Cheers.
Bye.